Welcome back, learners. Hope you all are doing good. From the thumbnail, you could get the clear idea of today's tutorial. So we are leading this video to the topic of DNA replication, which is the process where DNA creates a replica or copy of itself during cell division, specifically in S, that is synthesis phase. So let's learn. Detailed process step by step, one may raise this question: why do cells need DNA replication? From unicellular to multicellular body needs to make new cells constantly through cell division. Therefore, genetic information, that is DNA, must be transferred into these newly formed cells. So, the parent DNA molecule undergoes the process of copying known as DNA replication, which need to happen before a cell can divide. It has been more than 50 years. The replicon model was originally proposed by Jacob and Brenner. I think the concept of origin of this process is still evolving. As E. coli genome is circular based on that configuration, few terms we need to learn before we will be discussing about the whole process. These are the terms we are going to know about each of them. At first, replicator is the initiation of replication is directed by a DNA sequence. In case of E. coli, the replicator is ORI-C. Next term is origin of replication, the specific region where the DNA double helix denature or melt into single strands. The third term is replication bubble. The locally denatured segments of DNA is called replication bubble. The fourth term is template strand, the segment of single strands in the replication bubble on which new strands are made in accordance with the complementary base pair rule. And the fifth one is very important, that is replication fork. When DNA untwists to expose the two single-stranded template strands for DNA replication, a Y-shaped structure formed. And the sixth one and the final one, that is bidirectional replication. When DNA untwists, as in a circular chromosome or replication starting within linear chromosome, there are two replication forks. Two Ys join together at their top to form a replication bubble. In many cases, each replication fork moves so that bidirectional replication occurs. Initiation of DNA replication spans 245 base pair of DNA and contains cluster of 3 copies of a 13BP 80 reach sequence and 4 copies of 9 BP sequence. For the initiation of replication, an initiator protein bind to the replicator, that is ORI-C region, and denature the 80 region. The E. coli initiator protein is DNA A, which is encoded by DNA A gene, which binds to the 9 base pair region in multiple copies and leading to the denaturing of the region with the 13 BP sequences. DNA helicases, that is encoded by DNA B gene, are loaded onto the DNA by DNA helicase loader proteins, which is known as DNA C and is encoded by DNA C gene. The helicases untwist the DNA in both directions from the origin of replication by breaking the hydrogen bonds between the bases. Doing this, the required energy comes from hydrolysis of ATP. Each DNA helicase recruits the enzyme DNA primase, which is encoded by DNA G gene and forming a complex called primose. It is important because DNA polymerase cannot initiate the synthesis of DNA strand. They can add nucleotides only to a pre-existing strand. That is which is a modified RNA polymerase synthesize a short RNA primer about 5 to 10 nucleotides, to which new nucleotides are added by DNA polymerase. This RNA primer will be removed later and replaced with DNA. At this point, bidirectional replication of DNA has begun. Here, we need to understand differences between template and primers. Primer is a short segment of nucleotides bound to the template strand. The primer acts as a substrate for DNA polymerase, which extends the primers and synthesizes new DNA strand, whereas the template is the one on which the new strand is synthesized according to complementary base pairing. Now, the process of separation of double-strand DNA to two single strands is called DNA denaturation or DNA melting. Single-strand DNA binding proteins binds to each single-stranded DNA, stabilizing them and preventing them reforming double-stranded DNA by complementary base pairing, a process called reannealing. The RNA primer made by DNA primase is at the 5' end of the new strand being synthesized on the bottom template strand. The DNA primase at the fork synthesizes another RNA primer, this one on the top template DNA strand. Each RNA primer is extended by the addition of DNA nucleotides by DNA polymerase III. The polymerase can synthesize DNA in the 5' to 3' direction only. Please watch our DNA polymerase video for better understanding. Moving forward, maintaining that polarity, DNA is made in opposition directions on the two template strands. The new strand being made in the same direction as the movement of replication fork is the leading strand, and the new strand being made in the direction opposite that of the movement of replication fork is the lagging strand. The leading strand needs a single RNA primer for its synthesis, whereas the lagging strand needs a series of primers. DNA gyrase, which is an enzyme of class DNA topoisomerase. I also made a video regarding this. Please watch. So moving forward to DNA replication procedure, that DNA gyrase relaxes the tension produced in the DNA ahead of the replication fork, and this tension is considerable because the replication fork rotates at about 3000 rpm. On the leading strand, template DNA polymerase III synthesizes the leading strand continuously toward the replication fork. On the lagging strand, template, a new initiation of DNA synthesis occurs. An RNA primer is synthesized by DNA primase. DNA polymerase III adds DNA to the RNA primase to make another DNA fragment. Because the DNA strand is synthesized continuously and lagging strand is synthesized in pieces which are called Okazaki fragments from its discoverers Reiji and Tsuneko Okazaki. DNA replication is as a whole occurs in a semi-discontinuous manner. Eventually, the Okazaki fragments are joined into a continuous DNA strand. DNA polymerase I and DNA ligase ensured this event. The steps are repeated until all the DNA is replicated. This process is quite interesting. We need to look into deep that the 3' end of the newer fragment is adjacent to the primer at the 5' end of the previously made fragments. DNA polymerase III leaves the newer DNA fragment and DNA polymerase binds. The DNA polymerase I then digests an RNA primer strand and extend the DNA strand. When DNA polymerase I has replaced all the RNA primer nucleotides with DNA nucleotides, a single-stranded nick. Nick is something like a point at which the sugar phosphate backbone between two adjacent nucleotides is unconnected. DNA ligase joins the two fragments, producing a longer DNA strand. So this is a simplified way to learn about DNA replication. I recommend this book for the easy version of DNA replication process, so you must check it. Hope you all like today's video. Please subscribe our channel and hit the bell icon. Do not forget to like and share.